My name is Wick Haxton. I'm chair of the Berkeley's physics department, and I've got the honor of welcoming you to the annual Emilio Segrè lecture. This lecture comes at a really special time for the university during the 150th anniversary year of the founding of the Berkeley campus and of the University of California system. 150 years ago, the state gave Berkeley a mission to provide instruction and thorough and complete education in all departments of science, literature, and art, industrial and professional pursuits. I think we can take pride in how well UC has fulfilled that mission. On its 150th birthday, Berkeley stands as the world's premier public university, combining outstanding scholarship with the goal of making education accessible to all. We compete with the world's best privates. Harvard, MIT, Stanford, and Chicago are among our peers. In the last ranking of world universities, we were second in physics behind MIT and first in the natural sciences overall. But we are more and distinct from many other universities. As part of California's remarkable system of higher education, we represent opportunity. In this year's New York Times College Access Index, which is a measure of which institutions are doing the most to advance economic opportunity, remarkably, 6 of the top 10 places are held by UC campuses, including Berkeley. The excellence of our physics department and our capacity to educate have been sustained despite loss of most of our state support in the last couple of decades, in part because of help from the department's friends and supporters, many of whom are attending tonight. So I wanted to start by thanking you for all your support over the past years. Past Emilio Segrè lecturers are a who's who of physics, including Hans Bethe, Masatoshi Koshiba, Bert Richter, Mildred Dresselhaus, Ray Weiss, who was the winner of last year's Nobel Prize in Physics, and Bob Bergeno, our former chancellor, who's here in the front row tonight. Emilio Segrè was born in Tivoli, Italy in January 1905. His father was an industrialist, and Emilio described his extended family as intellectuals, academics, lawyers, and engineers. As a young man, he entered the engineering school of the University of Rome, but soon switched to physics, where he befriended Ettore Majorana and Edoardo Amaldi. He was tutored by Rossetti in experiment and by Enrico Fermi in theory, under the paternal oversight of the Institute's director, Corbino. Segrè received his doctorate under Fermi in July of 1928, and after 2 years in the military, he returned to the Institute, and in 1930 as Corbino's assistant, doing important work on the Zeeman effect in alkali atoms. He then moved to Amsterdam to work in Peter Zeeman's laboratory, and later to Hamburg to join Otto Stern. In 1935, Segrè became professor of physics at Palermo. The next year, he married his wife, and in the following year, Claudio, the first of their 3 children, was born. Shortly after they were married, the couple made their first visit to us, stopping at Berkeley's Lawrence Radiation Laboratory. Ernest Lawrence allowed Segrè to take discarded activated parts of Lawrence's cyclotron back to Palermo. Working with his Palermo colleague, Carlo Perrier, Segrè succeeded in extracting from the metal the element technetium, which has no stable isotopes. This was the first isolation of an artificial element. 15 years later, technetium was discovered in stellar atmospheres, proving that certain stars must continually synthesize heavy elements. In the summer of 1938, Segrè returned alone to Berkeley to continue his work. While he was in the US, Mussolini's government passed laws that barred Segrè and other Jews from universities. Lawrence hired Segrè as a research associate, and Segrè managed to bring his wife and young son to Berkeley. With the war depleting Berkeley's faculty ranks, Segrè became a lecturer in Berkeley's physics department in 1941. In 1943, he accepted Oppenheimer's invitation to join the Manhattan Project at Los Alamos, where he headed a group measuring fission rates. He returned to Berkeley as a full professor in 1946, setting up a research group to study nucleon-nucleon scattering, using a series of ever more powerful RadLab cyclotrons. In 1955, Lawrence and his colleagues designed the Bevitron to produce 6 GeV protons and energy sufficient to produce proton-antiproton pairs in collisions with a stationary target. Later that year, the discovery of the proton's antiparticle was announced in a paper by Chamberlain, Segrè, Weigand, and Ypsilantis. Chamberlain and Segrè were awarded the Nobel Prize for this discovery in 1959. He continued teaching and researching at Berkeley to and beyond his retirement in 1972, including a stint as the chair of the department. Over his 5-decade association with Berkeley, he helped train 30 PhD students, including C.S. Wu, Herb York, Tom Ypsilantis, and Herb Steiner. Emilio Segrè died from a heart attack at the age of 84 while walking near his home in Lafayette. Now, it is my great pleasure to introduce one of our department's most distinguished members, Emeritus Professor Ron Shen, who is uniquely qualified to introduce today's eminent speaker.
Good evening, ladies and gentlemen. I have the honor and pleasure today to introduce the speakers, Xiaowei from Harvard, not because I know her work very well, but because I was her thesis advisor. Well, here is a photo of Xiaowei taken more than 10 years ago, but she still looks the same. Well, many of you know Xiaowei's work much better than I do, and you can find description about her on the web. I would say something that you cannot find on the web. Xiaowei graduated at the top of her class from the University of Science and Technology in China at the age of 19. That's in 1991. I happened to be the chair of the admission committee of our department at that time. And when we received a recommendation letter from the president of her university, we naturally put her on our most wanted list. And she came and passed all the exams with flying colors. And she then wrote to Professor C.N. Yan or Frank Yan for advice. And Yan told her not to be a theorist, but be an experimentalist. Okay. I was lucky to have her join my group for PhD, working on nonlinear optical spectroscopy for soft condensed matter. Towards the end of her PhD, Xiaowei decided to test the job market. She applied to Kent State University. Upon her visit, the colleagues there told her that they would consider only a postdoc appointment for her. But after hearing her seminar, they offered her the assistant professorship anyway. Xiaowei then applied to Stanford for an assistant professor position. The recruiting committee there has split opinions. Half of the committee felt she should be appointed, and the other half felt that she was too young and had no postdoc experience. They finally compromised and offered her a prestigious postdoctoral fellowship that allowed her to work whoever she would like to. She took the fellowship and considered working with either Yoshi Yamamoto on mesoscopic quantum optics or Steve Chu on biophysics. At that time, she knew the basics of quantum optics, but could not tell the difference between DNA and RNA. But she was brave enough to finally decide on working with Steve Chu, and she became a rebel of no return. The second year, she joined Steve Chu's group Steve got the Nobel Prize. At Stanford, life was obviously tougher than in Berkeley for Xiaowei. I asked her, how's life at Stanford? She replied, very busy and tough. I have to do both research and write proposal for Steve Chu. I would like to have such a postdoc. Xiaowei then went to Harvard as an assistant professor in 2001 and was promoted to full professor in 2006. In the first few years at Harvard, she received essentially all the prestigious awards for young scientists. And in the ensuing years, many other prestigious honors and prizes. She told me once that she was now under real high pressure all the time, because whenever she visited her parents, they were asked, what new prizes you have received recently, and what other awards you're expecting. You see how the Chinese tiger parents would work on their daughter. Xiaowei, however, has had no difficulty to bear the pressure throughout these years. She has received many, many honors and prizes, too many for me to enumerate here. Just 2 months ago, she was awarded the Heineken Prize from the Dutch Academy of Arts and Sciences, and last Saturday, the Breakthrough Prize in Life Science. Xiaowei's research aims at developing imaging methods and applying them to problems of biological interest. She is best known for her work on single molecule imaging to study biomolecules and single virus cell interactions. Development of super resolution microscopy called STORM and its applications that led to the discovery of novel important cellular structures. And recently, invention of another novel single-cell transcriptome imaging method called MERFISH that enables quantitative mapping of thousands of individual RNA species in single cells, allowing system-level analysis of gene networks with molecular scale resolution. The last two topics are what she will enlighten us today. Please join me welcoming Xiaowei and congratulating her winning the Breakthrough Prize.
Wow. First, I have to say I'm really embarrassed by this fantastic introduction that Ron just gave, and feels really special to hear praises from your PhD advisors. So, all right, it's really a great pleasure to give this distinguished lecture in honor of an iconic figure in physics, Emilio Segrè. In fact, it's for me always a great pleasure to come back to Berkeley. This is a place where I had lots of fun memories, great mentors, and I just mentioned my thesis advisor, Ron Shen, and I also was so happy to meet my academic advisor, John Clark, who taught me how to choose classes and showed me all the greatness of Berkeley. And also, over the years, I had built many great friendships here at Berkeley. So it's a special place for me. And in particular, as many of you know, not just my PhD advisor, Ron Shen, but also my postdoc advisor, Steve Chu, he has deep ties to Berkeley too. So Ron, it's so remarkable that you've been a professor here for more than 50 years. So, I mean, I feel like Berkeley has to be a really great place to keep you for so many years here. And another thing I found amazing about about Ron, which when I just heard his introduction, then I felt like I am really his PhD student, because even our comments are similar, because ever since I met Ron in 1991, he hasn't aged a bit. He just looked the same. So I'll show you the visual proof of that. You know, this is what Ron looked like. I mean, I didn't have a 1991 picture, but that's close enough, 1993. And then if you look at him today, isn't that the same? And while actually I disagree with his comment, you know, I actually aged quite a bit. So then I felt like must be another special thing about Berkeley, and that is keep you young. Either that or it's the mystery of biology, which alert me to the dark side, you know. So as I mentioned that my postdoc advisor, Steve Chu, also has strong close ties to Berkeley. Although I did my postdoc with him at Stanford, he himself was a Berkeley graduate. And all these years, he told me that he really just loved this place so much. And 30 years after that, his PhD, he came back here to be a professor at Berkeley and a director of the Lawrence Berkeley National Laboratory before he got recruited by Obama to be the Energy Secretary. And several years ago, he gave the Segrè lecture. So, but I also want to admit that it always feels a bit different to give a talk at Berkeley. With all my, many of my teachers still here listening to my talk, I feel like I've been exempt, you know, in particular Ron Shen, you know, he's not only taught me how to do research. I mean, before I started in his lab, I knew nothing about research. So he really taught me how to do research, but all these years have been like a father to me. So imagine having your father sitting there listening to your lecture, not only a father, but a critical father, I have to say. So I still distinctly remember this thing. You know, when I wrote my first paper with Ron, and I tried very hard, and then I gave him a manuscript. And I knew what to expect, because I've seen what kind of things my lab mates receive backwards. Those days, we don't have, Microsoft Word to give you track changes and so on. It was all, printed out in a paper. Of course, it has margins. It has narrow space in between lines, and it has the back page to be blank. And then you would see Ron's red writing everywhere, filled up like a completely. You couldn't find an empty space there. So I was expecting that kind of a treatment too, because I knew how critical he is on papers. So then a few days after I sent him I gave him my manuscript, he called me into his office. And I saw this completely clean manuscript on his desk. I said, wow, I must be special, you know. My paper was written so well that Ron, I didn't really believe that. So then Ron sort of paused a little bit, and then I said, Xiaowei, let me tell you something. This manuscript needs a complete rewrite. So, of course, he didn't just stop there. You know, he taught me how to properly write a scientific paper. So after several rounds between me and him, and we were rewriting, it was quite amazing that we sent it to physical review letters, and it was accepted just like that, without the request of any revision. I have to say that I've never had any of my own papers that got accepted without any revisions. So Ron, I hope at the end of this lecture you don't tell me that you have to completely redo this lecture. So let me start with a little bit of a basics, because as Ron said, when I first started doing biological research when I was at Stanford, I really knew so little, and I really didn't know the difference between DNA and RNA. So I wanted to just give some basics, and because I know there are a lot of physics and even general public audience here. So what I want to share with you today is, as Ron said, two categories of imaging approaches that we developed that allow us to visualize the molecular world of life with high resolution, and also some of the biological discoveries we've made with those methods. So we know that biological systems, including human being, are made of this fundamental unit of life, which are cells. And then the cells are made of many, many, many different kinds of molecules. And here I give you some length scales. The cells are about 110 to 100 micron in size. And in thinking about how small it is, it's about just the width of a human hair. And then molecules are about 1 to 10 nanometers in size. So if you want to visualize the interaction of these molecules, how do they form intricate networks to give life collectively to a cell? And if you want to do it with a direct visualization approach and then want to think about how difficult that task might be, so then I want to give you an analogy. Let's sort of enlarge the human size to the whole Earth. Then the size of a cell is like the size equivalently of a Berkeley campus. And then the size of a molecule is like something in this picture. And who care to give a guess? It's actually like the head of a human. So basically, if you imagine yourself to peek inside living systems to see these molecules, it's as if you are like an astronomer looking into a distant planet with a very powerful telescope, and you not only need to see that planet, but you want to see the head of whatever life form is on that planet. So maybe not that difficult. It's a little exaggerating that way. So I'll give you, so then, if we are looking at these molecular interactions inside the cell, so what do we want for our visualization methods? I just mentioned the size scale. So we really like to have this molecular scale resolution. But also because there are many, many different types of molecules inside the cell. So we would also like to have the molecular specificity in our images to know the identity of these molecules. And of course, we're dealing with living systems which are changing and moving all the time. So we would also like to be able to do dynamic imaging to catch these dynamics. And if you consider these three requirements and think about imaging approaches that do that well, and you quickly think about light microscopy. In particular for these two reasons, molecular specificity and dynamic imaging. The latter one is easy to understand. We all know light is relatively gentle, and hence you could actually image living systems without worrying. I mean, you do have to worry about perturbation, but it's possible to achieve light microscopy imaging without perturbing the living systems. And also it has reasonably fast time resolution, so you could catch these processes in real time. And molecular specificity is for two reasons. One is, light is wonderful. I loved light or research related to light ever since I joined Ron's lab. Light has so many different colors. And also there are so many different colored fluorescent probes that can be linked to your biological molecule of interest with high specificity. For example, there are naturally existing fluorescent proteins in jellyfish that that can not only fluoresce, but also you can link to proteins in the using genetic approaches. Also, we all know antibody is not only good for fighting off pathogens in our body. You could use dye-labeled antibody to specifically recognize proteins in your body and so on. So these give us the molecular specificity. Used to be that molecular scale resolution is not something that the light microscopy has. And that's one major topic of my lecture today. And the reason that it used to be that light microscopy did not reach the molecular scale resolution is because of a well-known phenomenon to physicists, that's diffraction. We all know waves diffract. This is showing water wave diffract. diffraction. And the light also is a wave, so it also diffracts. So when it converts to a microscopy term, that means no matter how powerful a lens you use to focus your light to a tiny spot, this spot nonetheless has a finite size that in the transverse dimension is about half of the wavelength of visible light if we use visible light for imaging. And that's about 203 nanometers. And along the optical longitudinal direction along the optical axis, it's even bigger. So that means no matter how small an object you're imaging, be it even a single molecule, its image will have a finite size of about a few hundred nanometers. Now, if you have two objects that are far apart, their images will not overlap, and you resolve them. But as you get them closer and closer and closer, eventually their images will merge into a single blob, and that's when you're no longer capable of resolving it. And that was recognized in the 1870s by Ernst Abbe, and hence this diffraction limit is also called the Abbe resolution limit. So then to recap, here is the resolution limit, and the molecules are a few nanometer in size. So when you're using this light microscopy to look at these molecules, you see a relatively blurry picture, and you would like to improve it by at least 1 to 2 orders of magnitude to directly visualize the molecular interactions inside cells. And now we know very well that this resolution limit has been overcome, and it has been overcome by actually not just one method, but multiple different types of methods. Optically, there are two major categories of approaches that overcome this diffraction limit. And one is using a spatially coordinated manner to separate molecular signals, or we can call it using patterned light illumination, or this pattern illumination-based approach. And the pioneering method in that category is the stimulated emission depletion microscopy, or STED, that is pioneered by Stefan Hell at Max Planck Institute in Göttingen, and then he later generalized to this concept resolved. And I usually, when I talk to biologists, I don't go into anything in details about this slide, but since a lot of physicists are there, and then many of you know what stimulated emission depletion is. So basically what you do is you have an excitation beam to excite molecule to fluoresce, but in the meantime, you have a donut beam that actually can stimulate emission of the molecule from the excited state back to the fluorescent state. And then only in the center of the STED donut beam, where the intensity is near zero, that's where the molecules can give off fluorescence. So with that, you get these image of individual molecules, or the point spread function of microscope, to be much smaller than the diffraction limit. And you can see the contrast in terms of resolution boost. Another pattern illumination-based super resolution imaging method is structure illumination microscopy. In its nonlinear or saturated form, also overcome the diffraction limit. And the second category, another category of the super resolution imaging methods are the single molecule based approaches that I would like to focus on. But I should say that optical waves is one way to overcome the diffraction limit. But if you want to get higher resolution effectively, you could even using non-optical approaches. For example, recently it was developed by Ed Boyden's lab that you can expand your sample physically, and then you can use diffraction limited optics to actually get higher resolution than you would have by looking at sample without expansion. So the single molecule-based approach, actually in the single molecule field, it's been known for a long while now that even though the image of a single molecule is diffraction limited, but you can pinpoint the center position of this image with very high precision, nanometer precision. It's almost like if you're looking at a mountain, and you can actually pinpoint the peak of the mountain position to be a much better precision than the width of the mountain. And that was, even back in the late 80s, demonstrated by Jeff Gillis microsheets and coworkers with bead, and then use that to actually track kinesin movement with nanometer precision. And also in the late 90s, W. Moner demonstrated that we could actually detect single fluorescent molecules. And then he did that with absorption, and later Michel Aure demonstrated that you could also do that with fluorescence. So then later on, it has been shown by Paul Sullivan's lab that then you can look at the single fluorescent dyes molecule image and then pinpoint its center position with nanometer precision. So with that, then they were actually able to watch the another motor protein, myosin, move, head over head or hand over hand. I'm sorry, hand over hand is the right term. Head over head sounds a little strange, as if something has two heads. Actually, myosin do have two heads. So that alone in itself is a very powerful approach that has been used for biological research. But it doesn't overcome the diffraction limit, because the diffraction limit, the fundamental problem of it is what I just said. When the molecules are too close, their images overlap. So then in order to overcome the diffraction limit, you have to solve this fundamental overlapping problem. So that's where we came in. And then this overlapping problem is much severe than just two molecules, because in a biological system, you could have millions of molecules that give you overlapping images. How do you separate them? So now, more than a decade ago, we and others had come up with the idea that we could take advantage of the other dimension. So these molecules, their images could overlap in space. But if we take advantage of the time dimension, then we can separate them in time so that in both space and time, they do not overlap. So what we do is at any time, we activate a subset of molecules so that their images are no longer spatially overlapping. We pinpoint their center position with high precision. And then we turn off these molecules and we turn on a different subset. And we iterate this thousands of times until we determined all the molecular positions in the field of view. And then with that, you can see that the image is no longer limited by diffraction or by how wide these single molecule images are, but by how precise you can localize these molecules. And what led us to this idea is, you have to, because it sounds kind of simple. You turn on a subset of molecules. How do you do that? It's actually non-trivial. And what led us to this idea is, prior to this, we actually discovered that some fluorescent dye molecules are photoswitchable. They have different physical conformations or different chemical state. And in one state, if you shine in light, they can fluoresce. In the other state, which we call dark state, even if you shine light on them, to excite them, they won't fluoresce. But they can be activated from dark state to fluorescent state by a different wavelength of light. And then because they're separated by a barrier, energy barrier, so if you use a weak enough light, then you can get a small fraction of them that go across this barrier into the fluorescent state. And that's how we can achieve this, what we call sparse activation of a subset of molecules. And because of that, you can see we don't control which subset. We say, small subset, 0.1%, but it's a stochastic subset. That can explain this complicated term, stochastic optical reconstruction microscopy. But the reality of why we came up with this name is because Mike Russ, the first author, is really good at playing with words. So we thought Storm is a powerful name. So he tried to coin that in a way to fit this name. But I should also say that, as some of you probably know, that similar methods has been independently developed and published nearly simultaneously by two other teams. One is led by Eric Betzig, and his similar approach is called PALM. And slightly later than the publication of PALM was Samuel has published a similar approach called FPALM. And here is basically applying storm to this real sample, activating a subset, pinpoint their center position, and then eventually you get this kind of image of a biological structure, and this thing is called microtubules inside the cell, and you don't need to know exactly what that is, but you can appreciate by putting it side by side with a conventional diffraction limited image, you can appreciate the resolution gain. And I can give you quantitatively how much gain that is soon after, because I want to quickly jump into another problem in super resolution imaging, and that is so far I've told you ways to get a super resolution image in 2 dimensions, but basically all biological structures are 3-dimensional because we live in a 3-dimension space. So we not only want to overcome the diffraction limit in 2 dimension or in XY, but also want to do that in the 3rd dimension, the Z dimension. So how do you do that? In the concept of storm or palm, it's conceptually deceivingly simple, because in addition to determining the XY position of the molecules, we just need to add the Z coordinate. Of course, it's easier said than done, because when you capture your image on a camera, it looks like a flat image. So how do you get the 3rd dimension information? In reality, it's actually not that hard. If you have played with microscopes, many of you probably realize this thing, or if you played with cameras, with lenses in and out of focus, because the images in focus are sharp, and when they're out of focus, they're blurry. At the single molecule, for single molecule images, that blurriness basically means the image looks darker and broader. So in other words, the molecules in different Z, the shape of their images encode their Z information. The problem of this is, of course, we all know that in focus, it's the local minimum. So the slope here is 0. And below and above focus, they look the same. So there's a degeneracy. So we actually used a very simple approach to overcome this problem, and that is we inserted a cylindrical lens into the detection path. And this is what a cylindrical lens looks like, because they only bend light it's only curved in 1 dimension, so it only bends light in 1 dimension. And if you don't know what it is, but if your eye vision thing has astigmatism, the glasses actually correcting that is cylindrical lens. So with that, we can actually turn individual molecules' images from spherical into ellipsoidal form, and that ellipticity depends very sensitively on the Z position. And with that, we can actually do all 3-dimension super resolution imaging here. The color codes the Z, and then we can take any part of it and rotate around, and then you can see that. And here's another thing. This is, again, microtubules inside the cell, and then here's another cellular structure where I put conventional image, 3D storm, again, color encoding Z, and a cross-section for you, again, to appreciate the dramatic resolution gain. Now, in this initial 3D super resolution imaging work, what we accomplished is a 10-fold improvement in resolution, as you can see, both in XY and in Z compared to diffraction limit. And of course, if you can see something with 10 times better clarity, you will be able to learn a lot of new things. But on the other hand, tens of nanometers is also not really at the molecular scale. So depends on whether you're a glass half full or a glass half empty person. You can decide to do one of 2 things. You say, this is great. I use it for biological discovery. Half of my lab loves that. And I also have a bunch of physicists who have very high standard, and then they say, well, we want to continue to improve that resolution. So I won't bore you with details, but to say that over the years, we improve it with either the development of new imaging schemes or the development of different molecular probes that are brighter, then indeed now for some systems we can get to this a few nanometer resolution, which is at the true molecular scale. And then indeed now that's 2 orders of magnitude better than the diffraction limit. And I should say that super resolution imaging is a rapidly growing field. So in addition to my lab, there are many other labs who has been constantly doing these kinds of improvement too. So, and one particular thing that's worth mentioning is Stefan Hell's new method called MinFlux actually gets to 1 to 2 nanometer resolution. And also, I said one of the major advantages of light microscopy is you could do that for imaging living systems, capture dynamics, and indeed we could do that. And then in this actually relatively old work, we already demonstrated 1/2 time resolution, and since then it has also been substantially improved. Then I want to focus the rest of the first half of the talk on biological discoveries, because I'm a pretty firm believer of if you invent a new method, no matter how cool it is, if it doesn't lead to scientific discovery, it's still nonetheless a useless method. So the good thing about STORM is it's based on fluorescence microscopy, and which is widely used for biological research. And so inherit this broad application range of light microscopy or fluorescence microscopy. So here I only show you a subset of our applications to show you the range of things that we could study that go from really simple systems to, for example, this is a bacteria, and then we can see every single individual ribosome, which is the factory that make the proteins inside the cell, and this is a nice collaboration with my friend and colleague Sunny Shi. And then this is another system which is quite simple, which is an ion channel in sperm tail, which we collaborated with David Clapham's lab to identify this remarkable linear domain organization that David's lab later on shown to be important actually for the hyperactivity of sperm. That ought to make somebody happy. So, all the way to complex systems, these are cellular structures. These are actually just brain tissue slices where we could look into both synaptic structures. Synapses are these junctions between neurons that are important for signal communication in our brain. We can see the subsynaptic structures, see the molecular architecture, and this is a nice collaboration with my friend and colleague Catherine Duloc at Harvard, and all the way to circuit level picture scale where we can see how neurons communicate with each other. And not only things in the cytoplasms, but also see how our DNA is captured, like you see in the telomere loop in collaboration with Titia DeLong's lab at Rockefeller University. And then more recently, we can see these chromatin domain structures. But I want to focus on this last one, which is for 2 reasons. One is a scientific reason, and one is a personal reason. The scientific reason is in the early days, so when I give a talk about STORM, and people say, oh, Xiaowei, that's great. You have the super clear picture, and you can clearly improve some knowledge. But is there, what's the true discovery? Can you really make a true discovery? So this actually is a structure that shows that with this substantially gained resolution, we could discover cellular structures that you didn't even know existed before. And the other reason is the first author of this paper, when he was a postdoc in my lab at the time, but now he's assistant professor at Berkeley, Ke Xu. So this has to do with actin. So in contrast, I want to show you what actin typically look like in a typical cell. It forms these networks of long filaments and give you the cytoskeletal structure that are important for many functions inside the cell. But when we looked into neurons, we see a dramatically different kind of a structure. In the neurons have these special processes. Dendrites is one kind, axon is another kind, and the signal from one nerve cell to the other is propagated along the axon and then transmitted to the dendrite of the receiving neuron. So this is actually how the brain, the nerve cells communicate signals, and then that's the basis basically for all brain functions, cognitions, and so on. Or I should say that maybe I'm a little bit too neurocentric. There are a lot of biologists who study non-neuronal cells in the brain, like glials and so on. They're also very important. So back to the neurons, when we looked in this axon, which is almost like the electric wire inside the brain, and we see this periodic band structure of actin in addition to the long filament. Actually, this band structure is the most prominent actin structure that we can see in axons. And this is a three-dimensional storm image with the color encoding the Z information. So these bands are actually, if you turn them around, they're these little rings. So what we saw are these little ring-like structures that wrap around the circumference of the axon right underneath the axonal membrane. And a reason why you cannot see this structure before is if you do the same image in the same field of view with diffraction limit optics, that's what you see. So basically, you completely obscured this structure, and that's because the period or periodicity, or the space between these bands are just below the diffraction limit. So. And then actually this 180 nanometer spacing also give us clue for what other molecules could be in the structure, because if you see these actin rings periodically spaced, then you couldn't help wondering whether there might be a molecular spacer that are connecting these actin rings. And then one kind of molecule, and if there is, the spacer has to satisfy two requirements, and then it is 180 nanometer long, and it should interact with actin. And a molecule that require these two, satisfy these two requirements is called spectrin. And the best we know about spectrin is actually in a different kind of cell, very different kind of cell called red blood cell. And underneath the red blood cell membrane is this kind of a triagonal or a hexagonal network kind of structure. And these are tetramers of spectrin, and they're indeed about 190 nanometer long, and then they indeed interact with actin. So that led us to this hypothesis that what we saw are these little actin filaments aligned into ring structure. And the reason they're periodically spaced is because they're connected by the spectrin tetramers. And of course, this hypothesis needs to be tested. And a good way to test that is with multicolor storm imaging, where we image both actin and spectrin. And imagine we can not only imaging spectrin, but also imaging the middle portion of spectrum, which we could recognize it was an antibody, then we would expect alternating green, magenta ring. That's exactly what we see, alternating green, magenta band. And later on, we found many other molecules on this structure. I'll just give you a few examples. These little actin filaments are capped, and then we saw the capping protein, and indeed the capping protein show alternating structure with the middle portion of the spectrum. And since this is a structure right underneath the membrane, so it also anchor or adapter proteins into periodic structure, and these adapter proteins then anchor functional membrane proteins such as ion channels into periodic structure, as shown here. So we give this structure a name, and the name might sound a little bit boring, membrane-associated periodic skeleton, MPS. But the reason for its boring name, it actually has a story behind it. So I think it was like maybe a year or two after we discovered this structure, and I was at a Welch Prize ceremony, and at the table sitting next to me is a very famous biologist called Joe Ghostin. As many of you probably have heard of Joe, and he and Mike Braun, discovered LDL. And so basically, then he asked me, what's new in your lab recently? And then I said, we discovered this periodic structure. And then he said, oh, Xiaowei, you have to give it a name, otherwise people don't remember that. And then I said, okay, well, in the lab, we call it periodic membrane skeleton, PMS. So that's why now I thought that might be too provocative of a name. So that's why I call it MPS. So then, here is now, I just fast forward a number of years with more studies. Now we see a more complete picture of this membrane skeleton in neurons, where in axons we see this periodic structure, and we also see this periodic ring structure in a substantial fraction of dendrites. And also Stefan Hell's lab also see that. And then in some other dendritic region, we see these hexagonal network, but that's more prominent in the cell body. We see this hexagonal network. And this is a really prevalent structure where we basically see it in every axons, not only the same type of axons, but axons in many different types of neurons. And it's also conserved across animal species, from simple species such as C. elegans to fly, and then where we originally discovered it is in mouse and rats, in rodents, and then we see it in human brain, human neurons too. And then let me end this part just with what kind of functional roles that can be played by this structure, and there are a lot that are yet to be discovered, and we think it actually plays multiple functional roles. I'll just share with you a few things. So, in looking at this structure, thinking that the actin ring is a rigid one and the spectrum is known to be flexible, then you couldn't help thinking about this vacuum hose kind of a structure. I have an even better demonstration. So if you have a rigid ring connected by a flexible material, you see, then you can bend and stretch it without breaking it. And this might be indeed what a structure like axon or some dendrites needs, because these can be really, really thin structure and very long. So it can be brittle, and they can be constantly subject to mechanical stress. So you don't want to break them. So you need a flexible yet durable skeletal structure like this to support the axons. And this is not just imagination. There's actually some experimental data pointing towards that. And that is, if you delete this key molecule spectrum in a little animal called C. elegans, and then the axons actually tend to spontaneously break. Interestingly, if you actually make these animals paralyzed so that they cannot move, but they can develop into full shape, the axons can actually grow into proper morphology, suggesting that the structure can give the axon its stability under mechanical stress. And the labs who have done that interesting experiment is Eric Jurgensen and Michael Bastiani lab. And that's one kind of function. Another kind, as I mentioned, is we discovered even in the very first paper and then in follow-up papers from my lab and other labs that this structure anchor functional membrane proteins, and these proteins are very important for a variety of signaling processes in axons. So we think this structure likely play important roles in signaling in axons and probably in dendrites too. And also, this is a structure that are important for the formation of some of the very important functional structures in axons, such as axon initial segment and nodes of Ranvier. And both are important for the action potential generation and propagation in axons. And then recently, the Matthew Raspin lab actually showed that if you disrupt the structure in animals, you can indeed cause widespread neurodegeneration and a variety of neurological diseases. And actually, mutations in components of this structure is found in people who have neurodegenerative diseases. Now, whether the structure is really linked to those diseases are still open questions that need to be tested. And even though I only have time to focus on some of our own work, and I want to emphasize again that super resolution field is a big field, rapidly growing now. And then here is a review article that we wrote very recently, which shows you a variety of other labs' work that not only showing you the kind of different methods, some of them I've already mentioned, STED, PALM, main flux expansion microscopy, and so on, but also a variety of different kind of biological knowledge that we can learn from that, not only the molecular structures interactions, but also the stoichiometry or molecular counting and the temporal dynamics. So our hope is that with the further development of these higher and higher resolution optical imaging methods, not only optical imaging methods, but also other imaging methods too, like electron microscopy, X-ray microscopy, and so on. We hope that someday we will have a comprehensive picture of the molecular architecture inside the cell unraveling in front of us, just like how we watch movies of these things. And of course, with that, we would need the molecular and submolecular scale resolution that needs to be routinely obtained in biological systems and to directly review molecular interactions. That's still a active area that needs a lot of research. But also we need high time resolution and long imaging time to monitor complete molecular processes in real time. And that's also a very active area of research. But finally, one thing that I want to emphasize again is inside our cells, there's not just one kind of molecule or two kinds or three kinds of molecules. It's actually many, many, many different kinds of molecules. And it would be great if we can see these molecules all simultaneously. In other words, we would like to have this genomic scale to visualize the collective actions of all molecules and see how they give rise to this emergent property of life. And we made some progress along that direction in recent years in this genomic scale imaging direction. So I want to spend the rest of this lecture on this. And rather than start with details how to do that, I want to give you our concept of how to do that. Because, genomic scale, we're talking about being able to image thousands, if not tens of thousands of different kinds of molecules. If you think about traditional fluorescence microscopy imaging, when you're trying to image multiple species, you use multicolor imaging. You do 3-color imaging to image the interaction of 3 different kinds of molecules. Now, when it gets to thousands to tens of thousands, we really cannot have this many different colors. Well, that's not the proper way of saying it to physicists. I usually say that in this irrigorous way, then people already accept that. But I think to physicists, I should say that there are so many different colors, but the probes all have their spectrum to be wide enough that you cannot discern so many different color probes. So you cannot actually have tens of thousands of different color imaging. So how do you overcome that problem? So several years ago, I came up with this idea, which, again, on the hindsight, is pretty simple, and that requires combinatorial labeling and sequential imaging. It might look a little abstract, but if I explain to you, it's really quite easy to understand. So we encode these genes with unique binary barcodes. Each gene have a different barcode. And then somehow, I'll explain to you later, we imprint this barcode onto these genes. Okay? And then we read out this barcode sequentially. In the first readout or first round of imaging, we label and image only those genes that first bit reads 1, but not 0. And then we turn the signal off, and then in the second round, we read out those genes. Their second bit reads 1, but not 0. And so on and so forth. So for example, this is the first bit imaging, and those genes was the first bit reads 1, turn on, you detect them, second round, third round, and so on. And at the end of the imaging process, you see your molecules in the cell all look like little dots, and each dot will have a binary barcode associated with it. And now I ask you the simple question, every one of you will have an answer. After n-bit imaging or n rounds of imaging, how many different kinds of molecules you can distinguish? Now, the real amazing thing there is it grows exponentially with n. So with a binary barcode, it's 2 to the n. Well, of course, with a ternary code, and then you can get 3 to the n's and so on. So that means even just 16-bit imaging or 16 rounds of imaging, I can image some 60,000 molecular species. And that is sufficient to cover the genomic scale. So with this idea, we first applied in the central dogma of biology to this middle molecule, RNA, which is the message that we know that our genetic information is encoded in DNA, and then it gets transcribed into RNA, and then RNA gets translated into protein. Now, why we apply it to RNA first, both have a technical reason, which I will review very soon, but also has a biological reason, and that is, it actually tells you that what kind of genes are expressed in our cells. You look at our body, we're actually made of many, many different kinds of cells. And they're different not primarily because they have different genome. By and large, different cells share similar, if not identical, genome. They're different because different genes are expressed at different level. And then the first level of expression is this RNA. So if you can image all RNAs, they can actually profile gene expression and not only give you a molecular basis of these different cell types and their behavior, but also give you a quantitative way of identify cell types, which I will get to later on. So now comes to practical things of, this is a great idea, but how do we imprint that? How do we read that out? So we use a powerful approach for the labeling and imaging, which is called single molecule FISH, which is pioneered by Rob Singer's lab, and then later on further divided by Singer lab, Ajahn Raj, Alexander Van Wittenard, and so on, into a really quantitative approach. So basically what it allows you is to identify, image individual RNA molecules of a species and count their number inside the cell. And the process relies on hybridization. That's what the FISH stands for, fluorescence in situ hybridization. And many of you probably know that in our body, DNA is a double helical structure formed by base pairs of nucleotide. And RNA is another. Now I know the difference between DNA and RNA. So RNA is another kind of a nucleic acid and have these bases. And in single molecule FISH, it's identified by complementary oligonucleotide that can base pair with a fluorescent dye attached. As I said, it's a powerful approach that allows you to identify a RNA species, image it at the single molecule level, and count it. And then with our idea of doing this massively parallel approach, we envision that it can be used for transcriptome level imaging or genome scale imaging of RNA. And here's what we do. So we use this FISH probe to identify RNA molecules by hybridization. And these probes all have readout sequences sticking out, depends on the barcode, meaning that if the barcode reads 1010, it will have readout sequence 1 but not 2, readout sequence 3 but not 4, and so on. And then we use hybridization to recognize these readout sequences sequentially. And then for 16-bit, you could do 16 rounds of imaging or 4-color imaging in four rounds. Now, when I had this idea, I presented to my lab. I got the whole lab excited. But then soon, we realized that the picture that I painted is just slightly too rosy to be true. And the problem is, you see, I skipped this part, the error-robust barcoding. The problem is error. We are experimentalists. The problem is always error. So when I said the single molecule FISH is a very accurate approach. If you use it to image one kind of RNA, you can get to like 95% or better accuracy, smaller than 5% error. But if one bit has 5% error, 2 bits will have 2 times 5% error. 16 bits, you can do the calculation yourself to see how severe that problem is. So then how do we overcome this problem? We overcome this problem actually by borrowing a trick that existed in a far distant field, and that's digital communications. Because there, all the signals are transmitted by binary barcodes 1010. And they prevent the error by using error-robust encoding. Or, in a nutshell, these error-robust barcodes means you don't use all possible barcodes. You use a subset of them so that in between valid barcodes, multiple errors has to occur. If one has a 5% chance, having 2 simultaneously occurring will have a smaller chance, and 3 even smaller. And then if the Hamming distance is 4, which is what we often use, that means 4 errors has to simultaneously occur in one message to misidentify as another kind of message. And that not only reduces the misidentification error rate, but also it gives another advantage. We can correct the error, because if one error actually occurred in one bit, we know more likely where it came from, because it's uniquely close to one valid barcode and 3 errors away, or at least 3 errors away from the other. And so then we multiplied this for fish, and then I skipped that details. So that's why we have this name, multiplexed error-robust fluorescence in situ hybridization in MERFISH. And for those of you who don't know what MERFISH is, but only know what mermaid is, it's the flip. Okay, so it's not a pretty picture as mermaid. So we actually demonstrated that experimentally that it actually works. You know, we can use that to image 100 genes, 1,000 genes. I mean, really hundreds to thousands of genes reach the transcriptome scale, and then it has really high accuracy. So I won't bore you with the details, but for those of you who are interested, you can read our original MERFISH paper. Now, let me just end the talk with the kind of applications, and then still just focus on one kind, but I want to mention a few. You know, you could imagine that this is a good approach to give us the spatial organization of these genes inside the cell at the transcriptome scale. Actually, these spatial organization of RNAs are truly important for a variety of cell functions, and then we've demonstrated this large-scale spatial organization imaging already in our initial MERFISH paper. Another thing is for some, I imagine there might be some biologists in the audience, actually in our body, our genome, the DNA is folded into very intricate three-dimensional structure. And this three-dimensional structure is very important for regulating gene expression. So it's a very active field now to actually figure out what the three-dimensional structure of DNA is in our cell. So we actually demonstrated that using the MERFISH-like approach, we could actually do the 3D organization of chromatin or DNA and chromosome in the nucleus. And the concept is just we don't use the MERFISH to identify RNAs. We use it to identify loci of DNA, and then we see where they are, and then we can connect that into the 3D structure. But I alluded to this, and this is what I will focus on as the application that I want to mention today is the spatial atlas of cells in our body. I mentioned that we are made of many different kinds of cells, and not only many different kinds, they're organized in really exquisite spatial organization. I always say that if these cells are different kinds, but they're randomly distributed, you could imagine what we look like. That doesn't look intelligent at all, right? So that means the spatial organization is really key, actually, to tissue function. So how do we do that? And I mentioned that one of the quantitative and systematic way of identify different types of cells is to quantify this gene expression profile, what kind of RNAs are expressed inside the cell. There are actually already powerful ways to do that, and the one important way is called single-cell RNA sequencing. It really allows you to profile the different kinds of RNAs, their quantities in cell, and allow you to identify different types of cells. But a requirement of single-cell RNA sequencing is you have to dissociate the cell from the tissue. Not only that, you also have to extract the RNA out of the cell, then subject it to a sequencing experiment. So it lost this very important spatial organization information. So with that, I want to show you that, how MERFISH can help on that front. And then, so that means we get back to the cellular level, and to give you some some amazing numbers. In our body, there are actually 30 to 40 trillion cells, and we really do not have a good understanding of what are the different types of cells in our body, and I already mentioned that their spatial organization is critical for the tissue function, and hence we want to use MERFISH to help solving this cell atlas of human or other model organism problem. So our initial application of that is applied to a particular brain region called the preoptic region in the hypothalamus. This is a really nice collaboration with my friend and colleague Catherine Dulac at Harvard MCB department. And this is, as I said, an important region because it controls or regulates many of these essential social behaviors and homeostatic functions such as sleep. I care about a lot because I have really bad sleeping problem. And it's also important for feeding and drinking and thermoregulation. And in terms of social behavior, it's important for parenting, you see. Oh, no, this is not parenting. This is parenting and mating. And then here's a sleep picture. So we actually combined both single-cell RNA-seq and MERFISH in the study because we realize complementarities between these approaches. For example, single-cell RNA-seq give us a genome-wide expression profiling. It actually help us to select the marker gene sets for MERFISH imaging. And we selected the marker gene sets based on both our biological knowledge of this region and single-cell RNA-seq, and then we use MERFISH to do transcriptome profiling, in situ identification of the cell types that give us spatially resolved information. And because it has high detection efficiency and sensitivity compared to single-cell RNA-seq, some of the genes that are expressed at very low level is actually difficult to get by single-cell RNA-seq, and we can get those, and they're actually important for cell type, both identification and function. And I'll get to the end of it showing you that because it's an imaging approach that gets in situ information, we can combine with activity imaging to show you the function of these cell types. So this is one field of view. If we don't turn on the identity of the genes, so this is all the individual molecules, just to show you our signal-to-noise ratio, but then of course we know their identity. You know, when all the different kinds of genes that we image, their identity are turned on, you know, computer can actually extract that information. Your eye probably cannot. So let me help you by pare it down a little bit to show you several canonical markers. And then you can see, oh, these cells have this red marker, which is an inhibitory neuronal marker. So they're inhibitory neurons. These blue cells have this excitatory neuronal markers, hence they're excitatory neurons and so on. So it really gives you this direct information on the cell type directly inside the tissue. And we imaged a much larger region and identified the different kinds of cells. And here only I use different color to give you different major cell classes. But I'm going to move this region from the anterior to the posterior of the preoptic region to show you how dramatically the spatial organization of these cells can change over, you know, into different locations. Isn't that remarkable? So these are just major cell classes, and then inside neuronal types, we actually have identified more than 70 different neuronal types. That I cannot do it in color and yet still help you visualize. So I don't know whether this is helpful. It probably is going too fast. You can see the spatial organization of the 40 different kinds of inhibitory neurons and the spatial organization of the 30 different kinds of excitatory neurons. And then we can use, as I mentioned, the imaging in combination with activity marker imaging to figure out their cell type. Let me just walk you through this slide. So this is a parenting experiment that we did. You can see this mice exhibiting parenting information. And then when that behavior is shown, it actually will turn on a kind of a gene called immediate early gene CFOS in the neuronal types that are activated. So we included that gene in our MERFISH imaging. You can see that I deliberately actually do not show you all the different colors of different genes, but put them all in blue so that you can see the red is CFOS, showing you these are the cells that are neurons neurons that are activated after parenting. And since we have a spectrum or a profile of the gene expression, we can actually show you the important genes that are activated in this type. And with that, I won't get into the details here, but just to say that we applied that to study parenting, mating, and aggression, and identify the neurons in this brain region that are important for parenting, mating, and aggression. So in other words, what we have is a molecular and spatial and functional cell atlas of a brain region, and I'm always amazed with how pretty it looked like. But actually, I guess in our brain, the cells do not have these colors. But then, in a different region, that looks so different. And if I blow up, it's a really amazingly beautiful picture. And again, I want to say that there are different approaches to address different kind of complementary approaches to address this kind of a problem. And I want to show you a recent work from Carl Dijkstra's lab, because other than multiplexed fish, another type of approach that can do in situ gene expression profiling is in situ sequencing. Okay, and then labs like Mats Nielsen's lab, George Church's lab, and Carl Dijkstra's lab have developed approaches in situ sequencing. And here's a beautiful recent paper from Carl Dijkstra's lab using their in situ sequencing approach called StarMap. And again, this is a cortical region of the brain to show you the spatial organization of the cells. So overall, in the end, what we really want to get to is a whole human cell atlas, which we want to know what are the types of cells that are present in our body, how are they spatially organized, what are their functions, and how do they communicate with each other, and importantly, how do they go wrong in diseases. And it's like this, again, if I use this as an analogy, if we look at people from afar, it looks like a globe, but we really want to zoom in to see the cellular details as if we can zoom in the Google map on the computer. So I often say that this is like to generating a Google Earth map of cells in our body. And our body is a sort of just saying representative, because we want to do that also for model animals, for plants, for different biological species. And of course, this is really a mega scale project that would require a collective effort of many researchers, and not just one kind of methods, but also other different kinds of methods, as even in our work, we've already shown the combination of sequencing-based approach and MERFISH imaging-based approach. And there are many other methods that will be helpful that I don't have time to get to, but hopefully was so what I wanted to say is it is very important, has already been recognized by many labs. So there is actually an ongoing global initiative called the Human Cell Atlas Initiative that is a collaboration of many labs around the world with a variety of methods in hoping to give us this Google Earth map of our cells in our body. So with that, let me conclude by really, because there are many generations of my students who have done the work, and then I have listed all of them here, but I'm going over time a little bit, so then I won't be able to mention them all individually, but I really want to say that it is their, persistence, their not sort of discouraged by momentary setbacks, their hard work, their strong motivation, their creativity, and that actually made these kinds of results possible. And as an advisor, I'm very fortunate to have this group of truly remarkable, talented young students and postdocs to have worked with them over the past 17 years at Harvard. And also we enjoy many collaborators, and the scientific world is getting increasingly collaborative and multidisciplinary. You really need people with complementary expertise to actually join forces together to solve problems. And I also listed my remarkable collaborators here, and it's a very rewarding process because because of the complementarity, we learn from them, they learn from us. And I was a physicist. How did I learn all this biology? It is from these remarkable collaborators. So I also want to take this chance to thank this, but also emphasizing the importance of collaboration and multidisciplinary approach. And also I would like to, for the specific work that I did, I would like to single out a collaborator. This brain cell atlas mapping of the pre-optic region is a collaboration with Catherine Dulack's lab at Harvard. And I would also like to thank people, agencies for generously funding my lab throughout these years, in particular Howard Hughes Medical Institute and National Institutes of Health. And thank you very much for your attention.
Thank you, Professor Zhuang. First, and then I have a question. Why don't we apply the technique we use in MERFISH to maybe achieve protein image in tissue level to see the difference?
Yeah, that's a very good question, because I've shown you how do we apply it to RNA and DNA, really not because protein is not important. It's remarkably important. And multiplexed imaging of protein is something that will be very important to do, and we are interested in that. Other labs have already some starting efforts, and the difficulty are compared to RNA imaging. There are probably more than two major type of difficulty, but let me share with you two. One is, the base pairing is a very good way of labeling RNA, DNA. So any gene, you could easily design oligonucleotide probes to get those probes. It's yet still not possible to have a label for every single protein with antibody, for example. Some proteins have good antibodies for, and some proteins don't. So in order to do that, we want for every single protein type to have this kind of label. Fluorescent protein is something that you could genetically link to protein, but it's not possible, at least not yet, to think about how to genetically link for every single protein inside your body with different kind of fluorescent protein, right? So the labeling is a major obstacle in protein imaging. And the other is, in order to get this very high multiplexing power, you see we resolve individual molecules so that we can get their barcode. And protein often exists at higher density inside the cell. Resolving individual proteins for every single kind of protein, it has a different level of challenge because of that high density. That, I think, there are ways we could imagine to overcome. Super resolution imaging, for example, is one kind. So.
What about we use an antibody to bring a barcode to each proteins?
Yes, that would be way, but you have to, as I said, the antibodies for every single protein have to exist, right? And unlike for every single RNA gene, you can just design a complementary oligos. Yes.
Since you have to make so many iterations of the molecules in order to get the final image, and especially if you wanted to image living systems, how do you keep the molecules from moving even just a couple of nanometers so that you can continue to get the proper resolution? Right.
So was that question, I imagine you're referring to STORM, right? Because in MERFISH, we haven't even tried to do that kind of imaging for live cell dynamic imaging. And primarily there is because these probes need to penetrate and get to the genes, and there to deliver them to live cells is difficult yet. But maybe there will be ways in the future. I never think there's, it's impossible. So for STORM, that's a very good question. And that is because we do have to iterate it, thousands of times. Do we obscure in terms of, the dynamic information? Any kind of a super resolution approach, when you get higher spatial resolution, you always take a hit on the time resolution. These two are sort of a compromise. So with that, super resolution imaging do tend to have slower time resolution than conventional resolution. That trade-off is a necessary trade-off. That said, it's not a zero-sum game. We can continuously improve the resolution. I've already shown you that we actually can get sub-second time resolution with STORM, and other super resolution imaging methods, some can even be faster. But with any kind of a method, you have to think about during that imaging time, be it 10 millisecond or 100 millisecond, whether the system have moved enough to actually obscure the structure. And all those things are things that we do consider when we get the dynamic imaging, just to see, when I say that we can have 20 nanometer resolution with sub-second time resolution, that means the systems that we studied have not changed its structure or moved by more than 20 nanometers when we were imaging it in that sub-second time scale. But overall, that is that's why I said the time resolution is an important direction of improvement for super resolution imaging.
One last question.
For looking at the different behaviors of the mice, like in parenting or mating, if you're not able to do that live, what's your plan for trying to understand how the gene expression profiles change depending on the behavior? Is it to try to freeze it in place or something else?
Yeah, so basically you can get that information already imprinted in the cell before you actually fix the brain in imaging, right? I've already shown you that we can actually identify which cells are activated after parenting. That is because we did have, you cannot have a dead mice, of course, exhibiting parenting behavior, right? So what we did is we have the mice exhibiting parenting behavior, and then in that there's a surge of the expression of this immediate early gene called CFOS. And then we sacrifice the mice, but those genes are already expressed there to allow us to identify which cells are activated. So there are other activity markers that you could also think about, and then you can image the mice live to see those activity markers, and then you can fix the brain and then do this kind of a multiplexed imaging to find out, this active neuron, what type it is and where it is, and so on.
As a follow-up, would you worry about the profiles changing when you do fix the mice?
That is a good question. So there are, in terms of using immediate early gene, actually there's, a long sort of history of research to have shown that this is a very good approach. And people have proven that, these situations are not messed up while you're fixing. In terms of other kind of activity markers, so when you are imaging the animal while it's live to see these activity markers such as calcium and so on, and then even after you rapidly freeze or fixed so that things are changed, but I already recorded that while it's live. I just need to then think about a solvable problem, that is the spatial registration of that image with landmarks to my fixed cell image to connect which cell actually was the cell that had the spark of calcium signal and so on.
Thank you for a wonderfully entertaining evening.
Thank you.